Hello, in this video we're going to be talking about telomeres. So let's not waste any of your time. Let's get right to it.
It's hard to breathe, but that's alright. Hush.
Now, when we're talking telomeres, we're talking about a very specific spot on a DNA strand, and it's kind of something that a lot of research has been done on. There's been research that shows that maybe there's a link to the length of the telomeres and Alzheimer's. So that's a possible explanation for what's happening there. But the thing to think about is that, like on the lagging strand, when the RNA primers are used to kind of jumpstart that, when it gets to the end, on that 3' end, because it likes to read 5 to 3, so when it starts in the middle, working its way to the end, that little section there, that's left empty, or no replication is done there, and that's the section that we're going to be looking at. Now, when we've looked at everything we've looked at so far, the colors red and blue have been used. Blue for the original parent, red for the new strand. And so when that new strand, that new single-sided DNA is made, as I mentioned in the previous slide, when we get to that 3' end, the polymerase doesn't replicate that section. That's the telomerase section, and so each time, since it doesn't replicate that area, that section, each time the RNA can lose, the DNA strand, it can lose, as it says there, 50 to 200 kind of base pairs, but that end is made up of a set of nucleotides, and you can see the letter combination over there. About 25,000 sections at the end is that TTAGGG section, and so if you take 25,000 sets of that times 6, you know that you're going to get, what is that, 15,000 sets of that. And so each time the very end isn't replicated, and so the cell loses a little bit of length, and that's part of one of the reasons why after 20-30 times the cell replicates its DNA, the cell dies, because the ends just get too short, or it gets too short. And that's the part that's been researched as a connection with Alzheimer's. Now, just to remind yourself how this process works, you've got that telomere section. It's being red going, and you can see the arrows in that direction. And when the RNA is put there, as it reads along, it's put there, but when it gets to that end, and you can see there in step 4 there on the diagram, there's a gap that's left there, and that's the portion of base pairs that the DNA strand loses, the new one, and it's the idea of the telomerase putting it on there, and that end, that telomere, being left uncovered. Well, that's our video over telomeres. When we come back next time, we're going to be looking at DNA repair. We'll see you then.